Assalamu alaikum. So in this lecture, what we'll do is continue talking about transcription with a focus on eukaryotic systems. Now, the thing is, we are living in a very exciting time. We are just scratching the surface of molecular biology. Things are not only complex, but things are quite sophisticated. And we are demolishing all of the previous knowledge that we had before. We're establishing new science and new possibilities. What's also interesting and fascinating is the advancement of technology. And this technology has enabled us to learn a lot of things. What's even more fascinating is the development of informatics. And that's why I keep on saying, yes, we need medicine, but we need informaticians, specifically bioinformaticians and medical informaticians. We need people who are experts in IT. If you are a medical student or a dental student and you have capabilities in informatics and IT and programming, please do not hesitate to switch to that field. It's really exciting. And I think that the future lies in bioinformatics and medical informatics. Let's talk about transcription in eukaryotic systems right now. So the thing is, this is sort of like the anatomy of a eukaryotic gene. Okay, so we'll talk about the following concepts. We'll talk about, of course, the promoter region. Okay, we'll talk about the transcription start site, the +1 site. But we will also talk about the exons, the concepts of exons and introns. We'll talk about the concept of splicing. We'll talk about the signals for termination. We'll talk about other regulatory regions like enhancers and silencers. We'll talk about complex proteins, and we'll talk about DNA looping. This is another fascinating area. We'll talk about RNA processing as well. So there are a lot of things to talk about in transcription in eukaryotic systems, specifically in humans. So first we'll talk about the RNA polymerases. So in bacteria, there is only one RNA polymerase, that's in E. coli. Okay, but in human cells, there are 3 polymerases, and these are known as RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Each one of them is specific for certain genes. So RNA polymerase 1 transcribes ribosomal RNA genes. RNA polymerase 3 is specific for transfer RNA genes as well as one of the ribosomal RNA genes. Now, RNA polymerase 3 is our focus. RNA polymerase 2 is our focus because it is the polymerase that is responsible for synthesizing messenger RNA molecules as well as microRNA molecules. So this is going to be our focus. So things are more complex in eukaryotic systems, okay? And help is needed. So the RNA polymerase 2 in human cells is not like the bacterial RNA polymerase. The RNA polymerase in bacteria can do a lot of things. It has many hands all over the place. It can unfold the double-stranded DNA. It can initiate transcription and RNA synthesis. It refolds DNA. It reforms double-stranded DNA once it's done. But yes, it does need help in one thing, and that is binding to the promoter with high affinity, finding the promoter with the help of sigma subunit. But the human RNA polymerase 2 specifically, and this is our focus, needs help. And this help comes from proteins known as general transcription factors. So they are general because they help the RNA polymerase 2 in transcribing many genes, non-specifically. Okay, so they are general. And they're known as TF2, meaning transcription factor 2, and then they are given a designation like transcription factor 2A. So this is a protein that does certain things. Transcription factor 2B, it does something else, and so on. Okay. So what do general transcription factors do? They do a lot of things. They help position the RNA polymerase correctly at the promoter. So they help the RNA polymerase find the promoter and bind to the promoter. So they function more like the sigma subunit. They also pull apart the two DNA strands, okay, creating the open promoter complex. Okay, so they function more like the DNA protein. Okay, they help the RNA polymerase in also unfold in unfolding the DNA as it moves forward. So they function like helicases. They also push the RNA polymerase forward to begin transcription. So they're like the engine of the RNA polymerase. So they do a lot of things. So let's talk about the promoter region of eukaryotic cells. So it's not like in bacteria where you have the promoter region and then you have the -10 and -35 regions. It's more complex in eukaryotic cells or in human cells. You have something known as the data box. And this data box is sort of similar to the -10 region that exists in bacterial DNA. Okay, so this is located a bit farther away relative to the data box in eukaryotic in prokaryotic cells, which is located in the -10 region. Here it's located in the region spanning -26 to -31. You also have a transcription factor 2B recognition element, BRE, that is located even farther away from the transcription initiation site. You have something known as the initiator as a promoter, and this is located within the transcription start site. So it covers the region from -2 to +4. Okay, so the RNA polymerase can sit on the initiator element, okay, and it starts transcription at the +1 site. And then you have something, a promoter region that exists a bit downstream, and it's known as the downstream promoter element. Okay, so remember, this is upstream and this is downstream. Okay, now any particular gene can have one or a combination of all of these elements or all of these promoters. So you can have a gene that has these two as a promoter. Okay, you can have another gene that would have the initiator and maybe the data box. Okay, so you can have different combinations for different combinations of promoters or sequences or elements for different genes. Now, what happens is you have the RNA polymerase 2 now binding to the promoter region. Okay, and then you have the formation of what is known as the pre-initiation complex. Again, it's a complex. When we say complex, it's a collection of proteins. And this complex constitutes the RNA polymerase as well as a number of general transcription factors. Notice where each one binds to. Okay, this is just an illustration. Okay, so you don't have to memorize exactly where they bind to. So that's the pre-initiation complex. So this complex forms on top of the promoter, and once it settles down, it starts transcribing genes. Now, in addition to the RNA polymerase binding site and all these regions that I told you about, there are other elements that are important, and these are known as the promoter proximal elements. Okay, and they are located farther away from the data box and the transcription start site. Now, these regions are important because they are gene specific. Okay, and so for example, remember when we talked about operons in prokaryotic cells. And I said that basically it's a cistron, it's a genetic unit where you have transcription of one messenger RNA, but different regions would produce different polypeptides or different proteins. They are different from each other, they have different functions, but they participate in the same mechanism, like, for example, synthesis of tryptophan, metabolism of lactose, and so on. Okay, so in eukaryotic cells, there are no operons. There is no such thing as an operon, okay, because genes are monocistronic. But in order to coordinate synthesis of genes that participate in the same pathway or mechanism, they are controlled not only by promoters, the RNA polymerase 2 transcription, the RNA polymerase 2 binding site, they are also controlled or regulated by what is known as promoter proximal elements. And they are located within 200 base pairs upstream of the transcription start site. Okay, so genes that participate in the same mechanism, for example, metabolism of lactose, several enzymes are needed. Okay, now the genes that produce these enzymes are controlled by the same promoter proximal element, that is the same region. Okay, so that when they are transcribed together. Now, even if they are located on different chromosomes. Okay, so we say that the promoter region right here, the classic promoter region, the RNA polymerase 2 binding site, is important for what we say basal expression. That is the basic expression of genes. Okay, so producing very little messenger RNA. But in the presence of promoter proximal elements and proteins that bind, regulatory proteins that bind to promoter proximal elements, you can have a lot of induction, strong induction of transcription. And this would be more obvious when we start talking about regulation of transcription. Okay, so for example, again, so operons, what you have in here is an operon that produces different enzymes. Okay, enzyme E, enzyme D, protein C, protein B, and enzyme A, let's say. Okay, so different proteins with different functions, including enzymes. Now, so you have transcription of one messenger RNA that produces all of these different proteins with different functions. Now, this is an operon. Now, in eukaryotic cells, there's no such thing. So you have the enzyme that is needed for enzymes that are needed for synthesis of tryptophan are located on different chromosomes, different regions, and you can have two on the same chromosome, but they are far away from each other. But in order to coordinate expression of all of these genes at the same time, they are controlled by the same promoter proximal elements. Okay, so whenever you have induction of one, you will have induction of expression of all of the other enzymes. So there is something known as tissue-specific transcription factors. Okay, so for example, all cells, all somatic cells, all cells that contain our genome contain the exact same DNA copy, except for germline cells, sperm and egg. Okay, so all of them have the same exact genome, except that they do not have the same transcription factors. And that is why you would have gene-specific, or you would have tissue-specific gene expression. So for example, you would have the same gene that exists in skin cells, liver cells, and brain cells, except that this gene is expressed moderately in skin cells it is expressed at very high levels in liver cells, and it is not transcribed whatsoever in brain cells because it's not needed. Okay, so in this case, you will have this activating protein or an activator. You will have it expressed in liver cells, inducing transcription, but you don't have this protein in skin cells. So the gene is expressed, but at moderate or even low levels, and the protein does not exist whatsoever in brain cells. But not only that, there is a repressor protein that prevents expression of this gene altogether. Okay, so that provides what is known as tissue-specific transcription. Now, in addition to the promoter proximal elements, there are other regions or elements that exist in chromosomes, and they also regulate gene-specific and they also regulate tissue-specific gene expression. Now, these are different than promoter proximal elements. They are called enhancers. Okay, so enhancers are activators overall. And these enhancers, there's something interesting about these enhancers, and that is they can be located really far away from where genes are located. So there can be huge distance between the gene and these enhancers. Okay, that's one. Now, these enhancers, something else about these enhancers is that if you change their location, so if you take an enhancer and you place it close to the gene, they can still function. If you remove it and you put it inside a gene, they can still function. If you remove them and you put them behind a gene or downstream of a gene, they can still function. If you flip them, they can still function. So let's say that the sequence of the enhancer goes in this direction and you flip it so that it goes in the opposite direction, they can still function. So how come they are still functional no matter what we do with them? Because there is something known as DNA looping, meaning that the DNA loops around and it can get close to the gene. Okay, so you will have a protein, a regulatory protein that binds to the enhancer, and the enhancer loops around. But the interaction does not occur between the enhancer binding protein and the transcription factors or the regulatory proteins that bind to the promoter proximal elements. Rather, this interaction is mediated by a protein, large protein known as the mediator. So you can have this interaction between all of these proteins that bind to different regulatory regions, including enhancers, with the pre-initiation complex, the transcription pre-initiation complex, and this is mediated by the mediator. Findings from the ENCODE project revealed that there are almost 1 million enhancers in the human genome. And that makes up about 10% of the human genome. And this is huge, by the way. So we still have so much to learn about how gene expression is regulated. Now, does that mean, by the way, that if you have an enhancer on one end of a chromosome, would it be able to regulate the expression of a gene on the other end of the chromosome? And the answer is no. It's not that simple. Scientists have found that the chromosome is actually divided into different domains or regions. Now, these domains are separated by sequences known as insulators. So you have the chromosome divided into different regions. So right here you have a domain, Here you have another domain, you have a third domain, you have a fourth domain, and so on. So you have discrete divisions of the chromosome. And these regions are known as topologically associating domains. And they make up something like loops. Now, as I said, you have insulators, and these insulators are DNA sequences, and they are the binding site of proteins known as cohesion and CTCF proteins. So you have these two proteins forming a complex, and they sort of like they restrict the localization of genes and their enhancers. So you would have an enhancer within a domain regulating the expression of genes within the same domain, not outside the domain. In addition to enhancers, there are also silencers. And they have the opposite function of enhancers. So enhancers bind to activators, and activators can interact with the pre-initiation complex, the RNA polymerase, the gene transcription factors, maybe the promoter proximal elements, altogether facilitating and activating gene expression. On the other hand, silencers are elements within the DNA. They interact with repressors, and these repressors, again, they help silencers loop around. They interact with the RNA polymerase and maybe the other proteins that are bound to the promoter proximal element. And instead of activating gene expression, what they do is that they repress gene expression. So they bind to the pre-initiation complex, preventing it from moving forward. Okay, so that's what they do. They repress expression of genes. Now, so again, anatomy of a human gene, you can see that you have the promoter region that includes, let's say, the Tata box. You can also have the initiator and all of the other types of promoters. You can have the promoter proximal elements. These are within 200 base pair upstream of the transcription start site. Okay, and then you can have the enhancer as well, regulatory proteins. Okay, the third type of the regulatory element within the not protein element within the DNA. And this enhancer can be placed anywhere near the gene, taking advantage of the feature of DNA known as DNA looping. It can also be flipped as well. Okay, and then you have the transcription start site, and transcription starts at the +1 site and it goes on to termination. So let's continue talking about transcription now. So let's talk about the stages, the 3 stages of transcription. Here we're talking about transcription initiation, elongation, and termination. So in terms of initiation, the first thing that happens is that you have a general transcription factor known as TF2D. That's the first one that binds to the promoter. And what it does is that it recruits the other proteins, the other general transcription factors, as well as the RNA polymerase 2, forming the pre-initiation complex. Now, one of these general transcription factors is a factor known as TF2H. Now, this TF2H actually functions as a helicase. Okay, and what it does is that once it binds to the pre-initiation complex, what it does is that it creates the open promoter complex. Okay, so this is the signal. Now, transcription factor 2H also has another important function. It functions as a kinase. Now, what is a kinase? A kinase is a class of enzymes, a type of enzymes that add phosphate groups to other proteins. Okay, so what it does is that we say that it phosphorylates other proteins. So again, a kinase is a type of enzymes that phosphorylates other proteins, that adds phosphate groups to other proteins. So what it does basically is that it takes a phosphate group from a molecule like ATP. Okay, it takes this phosphate and puts it on other proteins. So TF2H is a kinase. Now what it does is that it phosphorylates the tail of the RNA polymerase. So the RNA polymerase itself has a tail, and this tail is phosphorylated. Once the tail is phosphorylated, the RNA polymerase is released from the pre-initiation complex. It's like the ignition, okay? And it starts moving, okay? So you have TF2H igniting the RNA polymerase to start moving and start transcription. So now you have elongation of RNA, synthesis of RNA, specifically messenger RNA, starting. So now how about termination of transcription? Now termination of transcription is determined by a consensus sequence, and this sequence exists in the messenger RNA. And the sequence is the following. It is AAU, AAA. This sequence is followed by CA, a dinucleotide, and then it is followed by a GU-rich sequence. Now what happens is that the RNA polymerase is just right here, right? It's getting to the end, of course, of the messenger RNA. So what happens is that this sequence is identified, or it's recognized by RNases, specific enzymes, and what they do is that they cleave the messenger RNA right after the CA. And it is followed by addition of a poly-A sequence. So the action or the whole process is known as polyadenylation. Okay, so you have this CA, and then this is followed by a sequence of about 200 A's. Now these 200 A's do not exist in the DNA template. Okay, rather it's not going to be A, it's going to be a sequence of T's, right? But they do not exist in the DNA template. So let's look at the DNA itself. What is the exact sequence in the DNA? I don't want to confuse you, but what we do is that we do not really, in molecular biology, we do not really show the sequence of the DNA template. We show the sequence of the complementary strand, the sequence of the sequence that is similar to the messenger RNA sequence. So we go from the 5' to the 3', but this is not the DNA strand that is read by the RNA polymerase. This is what we do in molecular biology. So I don't want to confuse you. So what you see here is not the sequence, it's not the DNA template, it is the complementary strand going from 5' to 3', because now the template, the DNA template actually goes from 3' to 5'. Okay, this is the complementary strand going from 5' to 3'. So the sequence in the strand that is read by the RNA polymerase goes like this. Okay, so you have to go in the opposite direction. So it is a it's a CA rich region right here, exactly. And here you have going from 5' to 3' again, it is TG, right? And the sequence is T, 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 A, T, T. But we use the sequence of the complementary strand that has again a similar sequence as the messenger RNA, except that there is a T instead of a U. In the messenger RNA, there is a there would be a T right here. Now, so this is how the messenger RNA looks like after termination and modification of the messenger RNA. Now, if we take this, by the way, this is just something on the side for now. If we take this and we want to convert it to DNA, which is a DNA that we call complementary DNA, it's called cDNA, and this is done using an enzyme known as reverse transcriptase. It's a viral enzyme. So what it does is that it converts it converts the messenger RNA to a cDNA molecule, to double-stranded DNA molecule. It would look like this. That's how it, okay, so it's double-stranded, so you have to, of course, you should know what the complementary strand looks like. Okay, so this is the messenger RNA after the end of termination and processing and modification of the messenger RNA. Now, so eukaryotic transcriptional units produce messenger RNAs that encode only one protein. And here we're talking about the mature, the final product of messenger RNA. It produces one single polypeptide. Note that I did not say protein. Okay, I said a polypeptide. Now this polypeptide can then have a three-dimensional structure with a certain function, and that's what we call a protein. Now this polypeptide, this one right here, this polypeptide can be cleaved into smaller peptides, and each one of them has its own function. And there is a pre-hormone, or I should say a pro-hormone, a hormone precursor. Okay, it's a polypeptide that is cleaved into shorter peptides, and each one of them has a function. Okay, so note that I did not say a protein. I said a polypeptide, and that's why we say that it is monocistronic. In addition, this polypeptide can be modified differently. It can be phosphorylated, it can be glycosylated, so you can have the formation of different protein isoforms. That's what we call them protein isoforms. So these are similar proteins with similar structure, and they have similar function as well, except that the activity of these proteins may differ. Some may be more active than others, or more stable than others. Okay. So we will cover this in the molecular biology course. So now we have something else that we need to talk about, and that is there are things known as introns. And these introns exist in the DNA template itself. Okay, and these are non-coding regions. They do not code for proteins. So what happens is that once, right here you have a gene that exists in a DNA, genomic DNA. And it is transcribed from the +1 site all the way to the termination sequence, termination signal, which is again, the AAUAA in the messenger RNA. It's cleaved, and you have the addition of the poly-A tail, the sequence of 200 A's or so. So this is known as the pre-mRNA. Okay, so it's not the final mRNA product. It's a pre-RNA, mRNA. So right here you have the messenger RNA containing the exons and the introns, and the introns must be removed. And they are removed by a special mechanism known as RNA splicing. So these introns are spliced out, and the exons are connected to each other. So right now you have the final mature messenger RNA that contains only exons with no introns. Now this messenger RNA again can be taken and it can be converted to cDNA using reverse transcriptase. Cells use this mature messenger RNA, also known as transcript, okay, to synthesize proteins in the process of translation. Okay, now, so RNA splicing is basically the process of removing introns out of pre-mRNA. So this is the pre-mRNA or the primary transcript. And this is the gene, of course. DNA would contain the sequences necessary to synthesize messenger RNA molecules or primary transcripts, pre-mRNAs containing both exons and introns. And then you have the process of splicing, whereby introns are removed and exons are connected to each other. Now you have the mature transcript. Now you have the final product of the messenger RNA. So what's the advantage of splicing? Now the advantage of splicing is that you can have something known as alternative splicing. And in alternative splicing, you have one gene with multiple exons and multiple introns, and these cells pick and choose what exons they want, producing what is known as protein isoforms. These protein isoforms have sort of like similar structure, they have similar function, but they are regulated differently and their efficiency may also differ. So for example, here you have a gene for a protein known as tropomyosin. Tropomyosin is an actin binding protein. Now what happens is that this tropomyosin is a large protein and it contains many exons. So right here the gene itself contains 14 exons. But not all of them are necessary for the function of tropomyosin in muscle cells. So tropomyosin is necessary for controlling muscle contraction. Okay, so it has to be regulated differently in different cells. So in smooth muscle cells, for example, contraction is different than striated muscle cells. Okay, so and that is why in smooth muscle cells you need exons 1, 2, right here, 4, 3 is not needed, 5, 6, and then 7 is not needed, so it's removed along with the introns. 8, 9, 10, and then 14, so you don't have 11, 12, and 13. So what cells do is that they take one, they connect it to 2, they remove the intron in between, and they connect 2 to 4. So they remove 3 along with the intron right before and right after. And then you have 4, 5, 6, and then they don't need 7. They say, I don't need 7. They take 8 instead. So they remove 7 with the introns right before and after. And then you have 9, 10, and then all the way to 14. So they don't need 11, 12, and 13. The function, the purpose of tropomyosin in smooth muscle cells is different. So they say, I don't want 11, 12, and 13. So they're removed. Now, on the other hand, striated muscles, on the other hand, take exons 1 and 3. They don't need 2, so they remove 2. And then they connect 3 to 4, 5, 6. They don't need 7 as well. Maybe 7 is needed in some other, cells like cardiac muscle cells. I don't know. And then they connect 8 to 9, 10, 11, 12, and they don't need 13 and 14. Okay, so cells can pick and choose exactly what they want according to their needs. So producing different proteins. So this is one answer for why we have about 21 or so thousand proteins similar to mice, similar to worms, similar to other organisms that are simpler than us, because what we do in our cells is that we take these 21,000 genes and we can make 100,000 different proteins and protein isoforms by picking and choosing what exons we need and what exons we do not need. So this note right here says that exons that are 3' to another exon are never placed 5' to it after splicing. So basically what it says is that exons, for example, 9 and 10 cannot be placed before exon 8. Okay, so they will stay in the same order, that is after exon 8. Same thing with exon 4, for example, cannot be placed right before exon 2. They have to be in that order. Okay. Now, so following the production of messenger RNA or during transcription as well, and after finishing transcription, messenger RNA molecules are modified. And there are 3 mechanisms of modification of messenger RNA molecules. The first one is known as capping. Capping is like putting a cap, a hat. Splicing, we already talked about it. Polyadenylation, we already talked about it. Okay, and the processing molecules, by the way, are bound to the tail of the RNA polymerase. So the RNA polymerase is a large protein and it has a tail. So and it moves like a train synthesizing messenger RNA. And when the tail is phosphorylated by transcription factor 2H, now, and it starts moving, synthesizing messenger RNA, what you have is these modifying or processing proteins. They jump onto the tail and they wait for the messenger RNA to show up. So once the messenger RNA gets out of the RNA polymerase, they jump onto the messenger RNA and they start modifying it one after the other. The first one that jumps on top of the messenger RNA, actually at the beginning of the messenger RNA, are the capping factors. So what is RNA capping or messenger RNA capping? Capping is basically the addition of a methylguanosine at the 5' end of the messenger RNA. So right here you have a messenger RNA, and right at the 5' end, the very beginning of the messenger RNA, the methylguanosine is added to the 5' end of the messenger RNA in a reverse orientation. So note that it's directed downward relative to the other nucleotides that exist in the messenger RNA. So this is nucleotide number 1 of the messenger RNA. This is number 2. This is number 3. Okay, and here you have the methylguanosine added to the 5' end, but it's added in a reverse orientation. This is the cap. So what is the importance of a cap? Well, the cap, adding this methylguanosine to the 5' end of messenger RNA, has many advantages. One, it stabilizes the messenger RNA, so it doesn't get degraded. Okay, so you have a lot of proteins that intend harm to messenger RNA, and they want to degrade the messenger RNA starting from the 5' end, but the presence of the cap protects the head, the beginning of the messenger RNA, so it prevents mRNA degradation, and it increases the stability of the messenger RNA. This is one. The second thing is that it signals the 5' end of eukaryotic mRNAs. And this helps cells recognize messenger RNA molecules from other RNA molecules, okay, so which are not capped. So ribosomal RNA molecules are not capped, transfer RNA molecules are not capped, microRNA molecules are not capped. So whenever cells see this cap on top, at the beginning of the messenger RNA, they know that it is a messenger RNA. Something else is that this capping helps recruiting the splicing and the polyadenylation factors onto the messenger RNA. So they recognize, so these factors recognize that this is a messenger RNA that needs splicing, that needs polyadenylation. Okay, just by looking at the cap. Something else is that the cap helps in exporting the messenger RNA to the cytoplasm. So what happens is that synthesis takes place in the nucleus, and processing also takes place in the nucleus. Okay, so after finishing messenger RNA processing, that is capping, polyadenylation, and splicing, you have other proteins that can recognize the messenger RNA, the processed final product of messenger RNA, and they take it outside of the nucleus to the cytoplasm so that it can be used for translation. So transcription takes place right in the nucleus, in eukaryotic cells, in human cells, and then it has to be exported outside of the nucleus. The other thing, the final thing about capping is that it helps cells and ribosomes in recognizing messenger RNA molecules in the cytoplasm, and these ribosomes, they bind to the cap and they say, aha, this is messenger RNA that has to be translated. So they bind to the cap and they start translating this messenger RNA into a polypeptide, and we will talk about it in the translation lecture. Okay. How about polyadenylation? We talked about it before. So we said that in the DNA you have this sequence AAT, AAA, then this is followed by CA, and it's followed by a GU rich GT rich sequence, right? There's a mistake right here. I should correct it. Okay. So that's a GT rich sequence in the DNA. Of course, this is not the strand that is read by the RNA polymerase. It's the complementary strand, right? Now, then you have the pre-messenger RNA. You have the exons and introns are removed, and then what happens is that this messenger RNA is cleaved right here after the CA, and you have the addition of the tail of A's, also known as the poly-A tail. This poly-A tail is added by a polymerase, an enzyme known as poly-A polymerase. So what it does is that it adds these 200 A's at the end of the messenger RNA. Note that the poly-A polymerase does not require a template to add this A tail or the poly-A tail. So the A's right here do not exist in the DNA template. So what is the significance of polyadenylation? It helps transporting messenger RNA out of the nucleus to the cytosol. Same as capping. So cells look at the cap and they look at the tail and they say, that's it. This messenger RNA is now fully processed. 2, it helps in translation, and we'll talk about it in the translation lecture. 3, the poly-A tail stabilizes the messenger RNA as well. So you have the capping at the 5' end, so these enzymes cannot degrade the messenger RNA from the 5' end. At the same time, you have the poly-A tail at the other end, and it also protects the messenger RNA from degradation. Then you have messenger RNA transport. Okay, so it gets transported out of the nucleus to the cytoplasm, and then it can be translated. In case you have a defective messenger RNA, that is a messenger RNA that is not polyadenylated, that is not capped, that is not spliced properly, then this messenger RNA cannot be transported out of the nucleus. Similarly, if you have a messenger RNA that is too large, it cannot be transported as well. So if there's a mutation that causes, let's say that there's a mutation in the poly A signal, okay, in this case this messenger RNA cannot be terminated. Its transcription continues until something happens and transcription stops, and in this case this messenger RNA cannot be transported to the cytosol. So everything is regulated, by the way. Now, what happens afterwards? Do messenger RNA molecules stay forever? No. There's something known as turnover. So molecules get degraded continuously. They have a certain half-life. So in bacteria, the half-life of messenger RNA molecules is 3 minutes. Okay, that's the average. Now in eukaryotic cells, they can last up to 10 hours, but the average is about 30 minutes. Okay, otherwise they get degraded. Now, how can they be degraded? Well, degradation can take place either from the 5' end or from the 3' end. Okay, so what happens is that you can have shortening of the poly A tail by deadenylase containing enzyme complex. So you have removal of the poly A tail, and that results in destabilization of the messenger RNA, and then it gets degraded by 3' to 5' exonucleases. So exo means like from the end, nuclease is degradation. So they start from the 3' end and they continue to the 5' end. The other mechanisms by which RNA molecules can be degraded is first you have what is known as decapping enzymes, or a decapping enzyme. It removes the cap and messenger RNA becomes unstable. This is followed by an exonuclease. Note that the direction by which it degrades messenger RNA molecules is from the 5' to the 3' end. So note the difference between this exonuclease and the other exonuclease that moves from the 3' to the 5' end. Okay, so let's talk about a phenomenon that exists in eukaryotic cells, and I want just to highlight this phenomenon. This phenomenon is known as gene amplification. And what happens here is that this gene right here, or let's say a portion of the chromosome, gets amplified. So instead of having double-stranded DNA in this region of the chromosome, rather you have this gene amplified. So you have multiple copies of the same gene on that location or that region of the chromosome. And this is known as chromosomal puffs. So it's almost like you have this person wearing a puffy coat, a coat that is, puffed up, okay? Or if you have someone who's muscular, this person is puffed up, he is a huge muscular, okay? منفوخ. So this phenomenon right here occurs in normal cells as well as in cancer cells. So this is a mechanism by which cancer cells can escape or can develop resistance from bacteria. I have to fix this, by the way, to escape chemotherapy, or to develop resistance, not to escape resistance, or to develop resistance from drugs like methotrexate. We'll talk about methotrexate in the biochemistry course. So basically, cancer cells or cancer patients are treated by giving them methotrexate, and methotrexate kills cancer cells, or cells that divide and proliferate or grow and divide. So what cancer cells do is that they amplify the target gene, or the gene of the target enzyme of methotrexate. So they amplify the gene, they produce a lot of protein from that gene, because now there are so many copies of that gene producing a lot of protein. And this is a mechanism by which cells can overcome methotrexate. Another target gene is a receptor known as the human epidermal growth factor receptor 2. This gene right here is amplified in breast cancer cells. And there is a type of breast cancer cells known as HER2 enriched. What happens is that in normal cells, this receptor is needed, and it helps cells to grow and divide, or again proliferate. But in cancer cells, the gene for HER2 is highly amplified. You have multiple copies of that gene producing a lot of receptor on the cell surface, and cells become quite sensitive to the presence of the growth factor that binds to the HER2 receptor. And when the growth factor binds to the receptor, you have activation or induction or stimulation of the signaling pathway starting from this receptor, and this signaling pathway results in cell proliferation. So cells keep on proliferating, forming a tumor. Eventually, these tumor cells become cancerous. Okay, and one treatment, by the way, for this type of breast cancer is by giving them an antagonist, that is an inhibitor of the HER2 receptor. So it binds to the receptor, and it prevents the receptor from functioning. Okay, and it is a quite successful treatment. So a simple gene in a human genome is basically a gene that would have one single promoter and then several exons, for example, and then producing one messenger RNA only, producing one single polypeptide. Okay, but remember that the number of proteins in our cells needs to be really high. So we need to increase diversity. We need to increase complexity as well, that would match the complexity of our physiology. And this happens via different ways. You know, one of them is alternative splicing, but there is a phenomenon that is quite interesting as well, which is you have a single exon, but there are multiple forms of this exon. Okay, so let's talk about this. So we have an interesting enzyme known as UGP glucuronosyl transferase. What this enzyme does is that it takes a sugar molecule, glucuronate, associated with UDP, a nucleotide. So right here you have the UDP, and this is the sugar molecule, glucuronate. And this enzyme attaches UDP glucuronate to a certain xenobiotic. What we mean by xenobiotic is something like a foreign molecule, like a drug, for example, a certain chemical. Okay, and it can also attach glucuronate to other endogenous compounds in our body. The purpose is to make them water soluble and to allow the body to eliminate these molecules easily, because if they're hydrophobic, if they are lipid soluble, it would be hard to eliminate these molecules from our body. So what this enzyme does, and this enzyme exists in the liver, is that it metabolizes, it changes, it modifies these xenobiotics, these chemicals, making them hydrophilic, making them more water soluble, so that they can be easily eliminated. So it has the ability, again, to take different types of molecules and making them hydrophilic. Now, the thing is, note that these molecules are really different from each other. So how can a single enzyme have different substrates with different structures? Usually enzymes are really specific to a certain substrates with specific structures, but this enzyme is really diverse in terms of the substrates that it can bind to. So how does it do that? Not only that, but the enzyme can also modify not only different substrates, different molecules, different chemicals, it's also present in different types of tissues. And there is some sort of like specificity in terms of not only tissue specificity of expression, but also the type of modification of these substrates and where they are modified. So this this table is not for you to memorize, just to show you the complexity of this enzyme. But first, let me just give you an example of how the enzyme looks like. So it's almost like a drill. Okay, so you have a single drill, but there are different flutes. So you can attach whatever flute that you want to this drill, and the drill would do the same exact thing. Okay, or you can have the same head but different hats. Well, it's the same head, but there are different hats, different functions, right? So what the enzyme does is that it does the exact same thing as a drill. Okay, it makes a hole in the wall, but by attaching different flutes, it can create different flutes. It can bind to different flutes, but it does the same exact thing. So the enzyme, it does exactly the same thing. That is, it can bind to different substrates, but it does the exact same thing, which is attachment of the glucuronate group or the moiety. Same thing with the heads. Okay, so you have the same head, but you can place different hats, different functions. So that's what the enzyme does. So how does it do that? Well, it turns out that the enzyme has two subunits. Okay, the first subunit is the catalytic subunit. That is the subunit, the part of the enzyme that attaches the glucuronate. But it has different substrate binding subunits with different forms and different structures and different shapes and different conformations. Okay, so same different words for the same meaning. So the catalytic subunit is encoded by 4 exons, and these are exons 2, 3, 4, and 5. On the other hand, the substrate binding subunit, the part of the enzyme that binds to the different substrates, is encoded by a single exon, which is exon 1. Okay? Now, and it turns out that exon 1 is really not a single exon. Rather, it is made of 9 different exons, and they're known as A1, A3, A4, A5. Do not memorize as well. So each one of them would form an exon 1 that would encode eventually a substrate binding site. Okay? Not only that, but each one of them has its own promoter. Each one of them has its own promoter. So if we zoom into exon 1, we would find that actually it is made of 9 different exons, and any one of these 9 can be spliced with exons 2, 3, 4, and 5, producing an enzyme with the different enzymes with the same catalytic subunit, but a different substrate binding site. So here you have exons 2, 3, 4, and 5, and here you have the different exons 1, and each one of them has its own promoter. So you can have different transcripts, different messenger RNA molecules. So you can have this exon conjugated or spliced with these exons, or you can have this other one, or you can have the third one, and so on. So each one of them would produce a different enzyme with the same catalytic domain, the same part that attaches the glucuronate to the substrate, but a different substrate binding domain or region. So this is how from a single gene you can produce different isoforms, different types of the same enzyme having the same action, that is attachment or conjugating glucuronate to different substrates, and it is produced in different tissues as well. That increases diversity. This is how we can have 20,000 genes producing 20,000 pre-mRNA molecules, but at the same time we can process them differently. We can increase diversity from the same gene, and we end up having 86 or 83,000 messenger RNA molecules producing different proteins. Here we have a third phenomenon. In addition to alternative splicing and having multiple exons of the same exon, that is, you have alternative polyadenylation. Now remember, polyadenylation is the process of terminating transcription in eukaryotic cells. So let's see. What we have here is the following. We have a gene right here producing a messenger RNA. That's the messenger RNA. This is before processing the messenger RNA. Here we have an exon. Okay, that's an intron. Here we have another exon right here. Then we have a poly A site right there. Then we have an exon as well in here. That's an intron. And here we have a second poly A site. We have a fourth exon. Then we have two types of poly A sites at the end. We have a proximal poly A site and we have a distal poly A site. We have a poly A site that is near the last exon, and we have one that is far away from the last exon. So as the messenger RNA moves outside, you would have the following types of processes that can take place. You can have, this is alternative polyadenylation right here. So you can have transcription terminating at the very first, at the proximal poly A site or at the distal poly A site, producing an untranslated region, a 3' untranslated region that is either short, truncated, or long, not truncated. Now, what's the reason behind that? Well, having a long 3' untranslated region would display a regulatory site, a site where the messenger RNA or translation can be regulated. More specifically, in this example, is regulation by microRNA, and we will talk about it in the following topic, regulation of transcription. Of course, you can have alternative splicing as well of the same messenger RNA. So you can have exon 1 with spliced with exon 2 or 3 producing different proteins. But here we have the same protein, but the messenger RNA is regulated differently, producing different levels of the same exact protein. So here in the next example, we'll be talking about how we can regulate messenger RNA stability. Now, remember, in general, if there is increased transcription, this means that you have higher level of messenger RNA, which means that you will produce more protein. And cells regulate how much protein there should be inside them. So if they need a lot of a certain protein, they would increase transcription, increase messenger RNA, and as a result, increase protein. If they want to counterbalance that, if they want to reduce the amount of protein inside a cell, what they would do is that they would decrease transcription, decrease messenger RNA, and as a result, decrease the protein level. Now, sometimes another way or another level of regulation is to keep transcription the same and regulate the stability of the messenger RNA. Or cells can do both, that is, reduce transcription and reduce messenger RNA stability to quickly reduce the amount of protein inside them. Okay, so there are different means by which cells can regulate how much protein they would have inside them. So let's talk about how cells can regulate messenger RNA stability, and I'll give you an example for that. The example is related to how much iron there is inside the body. Now, iron is really essential for the body. It's important for oxygen transport by binding to heme, which is part of hemoglobin that transports oxygen, and iron is important for enzyme function as well. We're not going to go into details about these, just know that iron is important for our body. Now the thing is, too much iron inside the body can be toxic because it would cause organ failure, cell death. As a result of cell death, it would increase bacterial infection. So it's really important to maintain a constant, sort of a constant concentration or level of iron inside the body. And that's what it does. Now, there are different molecules that play a role in regulating or balancing the amount of iron inside the body. I'm going to give you an example of two of these proteins that have sort of like opposite functions or opposite purposes, I should say. The first one is known as the ferritin. Ferritin is a protein that exists in liver, and its main purpose is to store iron whenever it is abundant. So if there's a lot of iron in the body, this iron is taken to the liver, it goes inside cells, and it binds to the protein ferritin. Okay, and ferritin can bind to 4,000 iron atoms, which is a lot. So the other protein is known as transferrin receptor. So whenever cells need iron, this iron is released from the liver. It's carried by a protein known as transferrin, and transferrin then binds to a receptor on the cell surface. It's taken up inside the cells, and iron is released, and the receptor goes back so that it can bind to another transferrin, and so on. So now, whenever the body needs iron, or I should say, whenever there is high level of iron inside in the body, the expression of ferritin, the level of ferritin should go up to store this iron, and transferrin receptor should go down because there is good amount of iron in the body, and vice versa. That is, the opposite is true, meaning that whenever cells need iron, ferritin level goes down and transferrin receptor on the cell surface should go up to increase the entry of iron inside cells. So it turns out that the messenger RNA of both ferritin and transferrin receptor contain a specific element known as the iron response element. Now remember, element, whenever I say element, it means it is a consensus sequence within DNA and in this case messenger RNA as well. So this region, this element right here, the iron response element, exists within the 5' untranslated region of the ferritin messenger RNA. And there are several of these elements at the 3' untranslated region of the transferrin receptor messenger RNA. So why is this element important? So what happens with these elements is that these are the binding sites of a protein known as iron regulatory protein. So this protein would bind to the element whenever there is no iron. So it would bind to the 5' untranslated region of the ferritin messenger RNA and the 3' untranslated region of the transferrin receptor. Now what happens is that if there is iron, then iron would bind to the iron regulatory protein. So this protein is sort of like a sensor, releasing it from the messenger RNA molecules. Okay. Now the consequences of whether the iron regulatory protein binds to the element or not is different between the ferritin messenger RNA and the transferrin receptor messenger RNA. Now you can read the slide, but I want to go to the next slide to explain it better. So if there is iron deficiency, if there is low level of iron in the body, the iron regulatory protein would bind to the 3' UTR of the transferrin receptor messenger RNA and the 5' UTR of the ferritin messenger RNA. What happens here in case of the transferrin receptor is the messenger RNA is that the messenger RNA would be stabilized. So it would have a long shelf life, sort of, okay? On the other hand, when it binds to the 5' UTR of the ferritin messenger RNA, translation is blocked. That is, there is no synthesis of the ferritin protein anymore, which makes sense because we don't need ferritin because there is no iron to store. But we do need transferrin receptor in peripheral cells to transport iron inside these cells. So when the transferrin receptor persists for a longer time, there would be production of higher level of the transferrin receptor to get iron inside, to get more iron inside cells. On the other hand, if there is iron overload, remember, iron is toxic if it exists at high levels. So if there is iron overload, if there is high level of iron in the body, in this case, iron would bind to the iron regulatory protein, releasing it from the 5' UTR of the ferritin messenger RNA, activating translation, producing more ferritin to store this iron. On the other hand, when iron binds to the iron regulatory protein and this protein is released from the 3' UTR of the transferrin receptor messenger RNA, the messenger RNA becomes unstable. It gets degraded, resulting in the production of less transferrin receptor. Again, there's iron overload. It's better not to get iron or to get more iron inside cells, otherwise these cells would die. I'm going to leave you with this slide as well. It explains the same process like I did. So you can just, read and enjoy.